in the light of all of what I've seen, I actually decided to kind of like change my talk quite a bit. So what I will do now is actually I will give you a blast of techniques that we do in the lab, and it will be a little bit on the expense of science, so many of the things I will not be able to go into depth, but like being here, I had the feeling that that's the best to just like show you what one can do and what we actually do in the lab. And basically everything that I show you today is based on microscopy. There is some of the examples, but I will then talk about this. So what we care about in the lab is really cell mechanics, and what we care about mostly is to also understand that from a physics perspective. And when I say that, I mean, I would like to really figure out how nature uses physical principles to perform different functions. And this is really what drives my research up and down, and you will see now many flavors of that. The first thing, let's see if that works here. Oops. That's super hand. So first I would like to really thank a couple of students that have done the work that I will present today. So half of them have already left the lab. Half of them, actually 2 are quite new, but already have some pretty cool results. So these are really the heroes of the stories that I will show you. And Khalil already did a great job in kind of like motivating that mechanics is something that if you care about biology, I think you also should start caring about this. And this was something very different when I started doing cell mechanics and biophysics like 25 years ago. At that time, like biologists kind of like patted me on the back saying like, yeah, you're a nice guy, but you know, like we don't really care. That has changed, and that's very nice. And these are just some examples. I think that by now everybody agrees that cell mechanics is really important to understand the biology also. You really need to include these things. For example, here, migration, proliferation, development. They're just some examples from the lab. Here's cell division, where we actually measure intracellular mechanics here. This is not from our lab. It's from Nelly Moore, from a Nature paper, where they actually show that here there's kind of like these rings, these intracellular rings that drive here actin. And that turns out to be a very beautiful mixing process here during the cell division to actually mix up all the content. And that's a nice collaboration with David Stramer in London, where we actually here see on the Drosophila embryo, we see here an actin is green and the nucleus is in red. You see hemocytes that are moving here over the surface and just cleaning this up. And this is just to kind of like give you an idea that mechanics is important and that with like microscopy, you can do a lot of cool things. And I don't have to tell you this, I guess. So the things that I really care about, and I tried to introduce this before, is that we have a situation that is quite annoying for us at the moment, because we know that we need to figure out what's going on inside the cell on the mechanical perspective. And here we're still lacking a lot of tools. And again, like this was already introduced several times before on this meeting. And there are many problems in there. It's like not just technically hard. Also, we have this kind of like this idea here with this flash. We have also activity involved, and that means we have driven processes, so we cannot use our toolbox of statistical mechanics. We really have to also come up with new methods here. And then there's this big field of cellular function, right? And of course, what we want to do, and what's the big goal of the lab, is to relate several cellular functions to the mechanical properties, and on the other hand, to see to which extent the mechanical properties are relevant for the function. And I really think that there's going back and forth, and that is a super exciting field actually to work in. Now, when I say like measure mechanics and that is complicated and all of this, like why is it so hard? Well, you might know this, right? You go to the shop and you would like to know if these tomatoes are fine, and what you do is to measure mechanics, right? You just basically, you touch it. So our sense of touch is quite good. We apply a force in the end of the day. We measure with our kind of like hands the deformation, and out of this we figure out if something is soft or stiff or highly viscous and things like that. So to actually measure mechanics, we have to also do this on the cellular level. So we basically need to get something like here a spring stiffness. We need something like a force and a displacement. And what we do in the lab here is using optical tweezers for this. So we have here optical forces that are driving here small particles around. For example, here you see that in live. That is here an MDCK cell. You can see how that guy here jiggles around. This is a phagocytosed particle that's inside the cell. And then what you can do is you basically look at the force you apply right here. And in red, you see the displacement of the particle. And you can already hopefully intuitively see, right, if the red curve would be very small, if there would be not a large amplitude, it would mean that the particle doesn't move a lot, so it would be very stiff. And on the other side, if you would have something that's more fluid, you would have a large phase delay here between this blue and the red curve. So a perfect Newtonian fluid, for example, would have here kind of like a 90-degree phase delay. So out of these measurements, we can get all the information we actually want. We can do this on an intracellular level. And this is really something that we have a lot of fun in the lab, and I can really invite everybody to try that. If you want, like, we can talk about this for hours, but I will not talk about this today at all, because this is not really microscopy, right? So what I decided on was switching gears and then really trying to see what can we actually learn by looking at the system from a microscopy point of view. And to understand a little bit why these things are so complicated, I brought you another little movie here. So what I have here is cornstarch. And this is like one of the days when my students were just kind of like mixing that on for me. And you can see I'm very happy, so I have a lot of fun here. And then when you hit that with a hammer, it actually behaves almost like a solid, right? So you cannot penetrate it. But when you give it some time, it behaves like a fluid. So this is really cool stuff. I mean, this is also, you can cook with it, of course, but you can also, for example, here I will try to shear it. And if you shear it quickly, it actually behaves like an elastic material. It can even form a little ball. And the moment when I stop, it actually starts to flow. Why do I show you this? Well, this is one of the things that makes cell mechanics actually so complicated in a sense, because it's not so simple. We cannot just simply talk about, oh, what is the stiffness or what is the viscosity? It depends on time scales. Here in this example, if I hit really quick, the system behaves more like a solid, right? And if I give the system time, a little bit more time, then actually starts to behave like a liquid. And having said that, you can ask yourself the question, okay, so I'm talking about biophysics. We're part of the biological world. So what are we? Are we a liquid or are we a solid? And I guess most of you would count yourself rather as a solid, right? Because we're not floating away. But to be strict, right, on long time scales, turns out that at least our skin, like when you eat a lot, you see this, right, behaves more like a liquid. And you can bring this to the extreme because like things start to flow if you pull on them for a long time. And extreme case is this example here, where you can actually here have really your skin pulling to the point where it's getting really large. And that's of course important for growing. If you're kind of like, I don't know, if you're pregnant, right, then your skin needs to grow and it also needs to flow back. So there's of course a big part of activity involved. We have cells turnover, so it's oversimplifying just to talk about a liquid. But it's, I think, kind of nice to really keep in mind that these things that are in our world very easy to describe turn out to be very quickly quite complicated on the cellular level. All right, so what the title was talking about was flows, for example, and I decided to actually share you with something here. This is a NG108, it's a neuronal-like cell, and that was the time pre-Life Act. Yeah, there was a time before Life Act, and this was my first task as I'm a physicist, right, as a student, to generate here the stable cell line. So I was impressively proud of this. But what you will see in a second is very nice. We can now look at a confocal microscope image here. This is the lamellopodia, and you see there's this beautiful kind of like flow. Now you can ask yourself the question if there is any chance to get kind of like out of these flows that we see here, because that's just now a deformation. Can we learn something about force generation in these systems? And what you need to do there is actually you need to first get flow fields, and this is just an overlay here. So we used here PIV, particle image velocimetry, which is just a nice method. There are plugins now that you can use to get here the flows from this. And now there's still the question, can we now get from these flows, in the end of the day, something like the forces that are driving it? I'm skipping now a lot. Like what you would need is you would need to know what is the stiffness and what are the viscoelastic properties. And then you can actually calculate this. We were making jokes about like showing some equations. So there's not a lot of equations, but at least this one I wanted to show. That's the simple version. That's basically not a tensor description. But what that tells you is like if you know the flow here, this U would be the strain, the deformation if you want. That's kind of like the strain rate, the velocity. We need here in this E, this is the relaxation modulus. We have all the mechanical properties involved. And then out of this, by integrating here over the history, you can actually calculate the stress in the system that would be explained by the flows that you see. And that is here actually another cell that I show here. It's just because here there's a little bit more dynamics going on, but you can use this actually, and then you can get ideas about the intracellular stresses that would explain here these flows. And these kind of works have been pioneered in the Danuta lab. And we also have actually, during my PhD, I've done quite something on this. So this is nice, because what that tells you is you can actually, if you know something about your system, if you know the viscoelastic properties, and you measure these flows, right? You can actually quite nicely estimate forces. You can have time dependent of the forces. And I think this is actually really a very nice combination between microscopy here and biophysics and cell mechanics. You can also then, this is also related a little bit to another field that was already mentioned very briefly by Khalil yesterday. It's attraction force microscopy. Just to show you that in a nutshell, what this does is basically you have a substrate that has, it's basically typically polyacrylamide, but it can be any elastic substrate. It has some particles in there, and if a cell sits on it and starts pulling, then it starts to deform this. So typically what you would have one image where the cell is either relaxed or you killed it, right? And another where the cell is pulling, and you can see here quite nicely how we have here these deformation fields. And out of these, you can then calculate later the strain fields and the forces. You can also do this in 3D. So this is a confocal cut here. This is, again, I'm going through the cell. This is actually an immune cell here, and you saw a little bit these dots that you saw there. These are just podosomes. And what you can see here quite nicely is actually that first it's dark, right? Then you see it, and here this darkish area that basically just means it's out of the plane, right? So the cell actually pushes down, and we can also get the forces in the C direction. So you can, yeah, these are the podosomes that we cared about here. So just, for example, here you can then get the strain fields in X, Y, and Z. And since we also have the C dimension here, this is often called 2.5D microscopy. And the reason is very simple. It's not really 3D because we're still on a 2D surface, but we actually measure something in the third dimension. That's why it's called 2.5D. Anyone can discuss that, but that's typically what's done in the field. And from that, you can then get traction stresses. You can calculate the stresses that the cells are applying on the substrate. It's similar to what I showed you before, but here it's much easier because the system is not viscoelastic. It's really just purely elastic. And you can also use these tools. It's something that was very nicely by Javi Trepat. You can get something like the internal stress. And this is something here. This is really just in a nutshell, all of this. So the idea is if we know the pulling forces, right, at different positions, we can figure out what's, doesn't work. Do you hear me? Okay. You can kind of like figure out, because like if the cell pulls here on the substrate and here too, then the stress needs to transmit it inside the cell. And that's what basically this is. Yeah, sorry, there are no scale bars. I just cut that out. Just to give you an idea. Yeah, and this was very fun yesterday, right? So I have to admit that I'm not only a MeFibio virgin, I also had no clue about oysters. And so thanks, Olivier. He actually showed me yesterday what happens when you put some, and you all know this, right? When you put a little bit of lemon juice on this, what happens is, tack. You basically just see it contracts, right? So you can now ask all kind of biological questions, why, which channel, all of these things. Or you can ask the very simple, naive biophysicist question that I'm asking here is like, what force does it pull? And in the end of the day, all I showed you so far is basically just the same thing. To answer that question, you would just have to quantify a little bit here the displacement, this flow. It looks a little bit like a lamellopodium here. If you would know the viscoelastic properties, you can use the same kind of physics to extract the forces. And if you're really good and you really know where like all of these displacements are, you have actually even spatially a good chance to know where at which moment these forces are applied. So I didn't go through all the math yesterday, but I wanted to share that with you. Okay, so the overall problem that we have here in these situations is always very similar, and I try to motivate this like we want to get forces from motion, from tissue that's in motion. And what we really like here, this is fun, you know, but in the end of the day, this is a serious thing, and it's really interesting if you look at more complicated organisms. And since we also have zebrafish here, like at the Mifobia, I thought like I also include this part. So what you have here, that's a movie by Don Kane, a beautiful dark field microscopy video. And you see here the cell divisions, and at some point you see the onset of collective migration here, right? So we see, bam, now it starts. We have this epiboly collective migration. Some cells actually go against the flow. So all of this moves down. It's just a beautiful thing. And I can tell you for a physicist, this is just like mind-blowing. I guess also for biologists, like that within a couple of hours, you already have something that looks like a fish. So as physicists, when you look at this, you don't want to go to the end part because it's crazily complicated, but already this early part, this first part of collective migration, this is what we started to wonder, and we wanted to see to which extent we can describe that motion by simple physics, right? So as a physicist, of course, you're always kind of guilty in trying to simplify the hell out of things, right? You want to make it as simple as possible. And then it's always very risky in biology that you oversimplify your system. So when Bernhard, this was actually my first student in Germany, when we saw these things, like we looked at this, the Peabody process, and I told him, okay, it would be cool to try starting to measure, to figure out what's going on in there from a biophysics perspective. He looked at it and he said, oh, it really looks a little bit like this. It really looks a bit like a drop wetting a surface. And I thought, okay, like even for me, that was a little bit too oversimplifying. So, but I mean, he's a student, right? You let them kind of like pursue their ideas. And I said, okay, let's see, like after a short time, he will come back and tell me like it's more complicated than this. But I mean, I let him go and it was actually a lot of fun. So he learned all the microscopy here. And then he started to looking at such a wetting process and try to adapt it here. And on a wetting process, you might recall what you need is actually the contact angle here. So what he did was he was using the light sheet that we had just at the time access to. So these were our first light sheet microscopy images. This is now quite late in this Peabody process. So typically we just use this early moment here. You can do the tracking of the cells. We have seen all of this beautiful data yesterday by Merlin. So it feels kind of like lame to show this here, but we can also do something. But actually I will start to try to copy what they have. Important part here, though, is that all the physics that we care about from a wetting perspective is hidden a little bit in this contact angle. And you can write down a very simple equation that relates the forces pulling here. These are just like the surface tensions on the system to understand what's going on. And you can write this down, and then what you need to do is you need to measure this, and that's what he did, basically. That's quite nice. You can find analytical solutions if you have this layer here that intersects with the York, and the blue one is the top layer. You can do a little bit of geometry, and then you actually have here a very nice measure of this angle. And what you do over time, you can see quite nicely that this is the cosine of the angle. The cosine starts to increase, and that's what you would expect, right? Because while it moves down, the angle goes down, right? So the cosine should go up. Okay, so far so good. So that's a kind of a curve. Then I spare you all the theory. So we actually came up with a quite simple, I mean, it looks maybe complicated, but it's an extremely simple theory. All that's in there is that we say there is a force between cells. There's a force between cell and York, and there is a change in proteins that changes over time. And that actually will affect here these forces. And this is everything that's in this little theory, and that already works like a charm. So you can actually fit quite well here this curve. And then, of course, like he was very, very happy and proud. And I hated to tell him, but you know, like this is biology. This is this one example that you made. Like you actually have now to measure more and more of these. And I was just not expecting that this will work all the time. So he went back to the lab, and these were like over, I think, like something like 10 to 15 embryos here. And you can already see like each of them are different. This is exactly what you know when you do biology, right? And as a physicist, very frustrating. These little critters, you know, they're all a little bit different. But still, we thought like maybe like all of them kind of like have somewhat the same behavior. And here the idea is that you rescale here the axis. That means the time scale you rescale based on what is here in this exponential function as a time. And here you have to also calculate out here a rescaling factor. And what happens then is you collapse, you kind of like apply this to all these curves, and then very nicely all of these collapse on a master curve. And this one is also very nicely fitted to this. So what do we learn here? What we learn here is actually that you can describe even so complicated situations like the zebrafish development by a very simple wetting process. And you can go deeper inside. You can now kind of like make out of these parameters that are in here hidden, you can quantify, for example, these interaction forces or time scales, also interaction forces between cells and York. So you can really get an idea. You can see that in every zebrafish that's a bit different. But in the end of the day, we always find a fish. And that's quite fascinating because it means the relation between these cells. And this is something, if you care about, you can read up in this paper. So this was really very, very cool because we could see that with wetting, we could already explain a lot of the things in the zebrafish. Now, Alejandro was the next student on this project. here we were asking the question, can we go a bit further, and can we kind of like, he was actually getting nicer images of this, and he was then also starting to really track very nicely these projects. Can we get maybe more information, maybe even on the force level that's acting in there? Because so far it was just the surface tension, it was not at the inside. Now, what we kind of like did there, we got all of these traces, and this is really similar to what we've seen yesterday. And then the idea is really to figure out a way to calculate now from these flow fields, which would be here actually these kind of like red lines, can we figure out something about the forces that are driving it? And to do this, you have to kind of like, work with equations like this. So for the physicists, this is nothing than a Navier-Stokes kind, an overdamped Navier-Stokes kind term here. And then we have an additional force term here that would have something like internal forces that are gradient of stresses that could, for example, be acting cells that just pull on each other. And you can also have external forces or surface forces. For example, that would be forces that act between the cells and the yolk. And these are the two forces we can kind of like plug in there. And the idea is then to kind of like solve this system to get a prediction for these forces. And that's really a master thesis actually of Leon that's also published. So what Leon did in the end out of all of these kind of like data and prediction, he came up with such a map to model this. So what you see here on the right side is just like the same data, if you want. On the right side, we see all of the little cells that we follow. In green, this is just an idea to give you an idea about the density, the thickness in this particular region. And here on the left, he has in blue, he has actually the velocity of these flows. And what you see there in orange are the predicted forces. And if I let this run, you can see that actually here at some point, there is quite a force component going upwards. And if you remember before what I showed you on this video, this is exactly when gastrulation happens, when the cells move upwards. So that's kind of nice. That means we can use these kind of theoretical approaches to make predictions for the forces. But you might say like, well, come on, that's really far-fetched because A, we assume it's a liquid. I think this is actually a good assumption. But B, we kind of claim to know the viscous properties of the system. This is something that's really hard to know. So to really understand what's going on here, what we need to do is we also need to kind of like measure the forces and compare it then to these predictions. And this is really the question that I want to address now. So we can predict such forces. That is already very nice. But can we also measure them, cross-check, and then on the long run, this might be also a way to measure, if you want, indirectly the viscoelastic properties, right? Okay, so what has been done there? How can we measure actually forces inside tissue? And there's a certain toolbox out there. For example, one that was really kind of like employed very successfully in the past. Here, this is from C.P. Heisenberg's lab. You can make cuts, and then you basically look how such a cut, for example, here, they cut it and it doesn't really open, but on this axis it opens more. And then the idea is that there is more mechanical stress in this direction. Another thing how you can measure forces or also properties was pioneered here by Karin Gevorkian. At the time, she was part of the François Brochard lab. And this is a micropipette you basically pull, and then the moment at which you start seeing here the material floating into the micropipette, that tells you something about the forces of the surface tension, and the speed how that flows and tells you something about the viscosity of the system. That's a very elegant and simple measurement. And another one that was pioneered really by Oja Campas at the time at Harvard are kind of like droplets. So these are just oil droplets that are also here in a zebrafish. It says epithelial cells, but these are just the epithelial cells of the zebrafish. And then you can basically see at the shape changes of this oil droplet, you can get an idea about the forces acting on it. And that's really something that was triggering us a bit, and we were trying to start work on this. And at the same time, a very nice paper from Monica here, Dolega, from Giovanni Capello's lab came out, where you actually have here elastic tension sensors. So the difference between this is an oil droplet, and these are just polyacrylamide beads that then you can actually follow, and you can take a look at the shape changes. So the nice thing about this is polyacrylamide, and these oils are pretty inert. You can really inject this into cells, into tissue. Normally there is not a big problem as long as they're not too large, and you can then follow that over time. You don't really disturb too much your organism, and you can get an idea about the forces. So we actually picked up this idea, and we also developed that in the lab. And it might come, yes. And this is just now here an image. You've already seen it before, maybe in these like images where we had these green dots, and these are actually these sensors. And you can already hopefully see, also, I zoom in here, this is one of these guys. Since we have light sheet microscopy, we have these kind of like nice 3D shapes of these. And then the problem boils down in getting out of these shapes the information about the stresses, right? And so we partially make these beads alone, but partially we actually had the luck that Jochen Guck actually could give us part of them. So that's him, that's Jochen, unfortunately, and that was really a big blow for the field. He died just 2 weeks ago. I don't know if you've heard about it, but that is really the master of like trying to bring also cell mechanics into clinician, into medicine. So it's a really big loss for the field. So he had actually had some beautiful microfluidics, and that is also allowing you to generate these particles. So his particles have always the same size. Ours are just like very variable. That's why we either use ours or his. So now you have such a shape. What do you do with this, right? And there are several ways what you can do with it. This is the underlying equation that you have to solve again. The U here is the displacement. These are basically the mechanical properties that are involved. And what we care about here are the surface forces. And this looks like an innocent little equation, but it's quite complicated because you have here all these second derivatives. And there are different ways to solve this in the field. One that is done most of the time, I would say, these are numerical approaches where you do finite elements. You can actually put this in the computer and the computer will spit you back what force is at which point. That takes quite some time. So Alejandro really was going on and he wanted to find an analytic approach here. The idea was that the analytic approach will be much quicker than the numerical approach. And also one nice thing about the analytic approach is that we get something like length scales involved into the different forces. Okay. So how does that work? If it goes on. So what we do is we actually put this into spherical harmonics here. And with these, we can then generate all kind of shapes or approximate these. We can then really look at this in the sense of, oh, yeah. I feel more like a star now. And then we actually get here these shapes, but also the forces acting on the surface. And here there's just two situations, a bit early and a bit late. And if you care about this, there's just a biophysical journal article about this, where he has pre-computed all these solutions in. And this is something you can then just put in Python and that works at home. So it has a segmentor in it and already calculates these forces. The nice thing about this approach compared to others, the others are nice, huh? There's no problem. The advantage of this one is just that you would have in different length scales automatically because of the spherical harmonics. All right, so what we now need to do is we need to combine these bead measurements with many of these embryos, but how can you register that? And this is also something that we try to solve. So the problem is each measurement, the embryos are a little bit different, right? So this might be one, right? And then what you can first do, you can easily register like what is the top if you want, right? That's quite easy. You have the center of the sphere, you have the center of mass of this part, and then you can rotate it. But there's still kind of like this azimuthal rotation because like there's a front and a back of the system. How can we do this? And that was kind of neat. What Alejandro really here showed was he was measuring now all the forces that are acting, and not the forces, the flows that are acting there. And what you see here is basically on this coordinate system, like that azimuthal line. And then you can see that in a certain direction, the velocity flows are kind of like positive. That just means they go in this direction, right? And in other regions, they're negative. And what you can actually see, there are regions where the system is converging here. That's called the shield region. And that means we have a switch from a positive, like here to the left or to the right, and a negative to the left direction. And this is something you can also see here. And when you actually now look at just the overlay of all of these curves, that would be something like this. This is nothing than like the sum of all of these flow velocities here. You can then say, okay, well, if we don't rotate, you might have one measurement like this, like this, like this. All of them are a bit different. But if you kind of like fit a sign to this, and then you can rotate this, you can all put them kind of like on that kind of line. And then the nice thing about this is we can now cross-check this idea because we added here something that's called, oh, this should be green, I'm sorry. Goosgoid, so this shows up here. This is a marker for the cell body in the late stage. So we kind of like know where the embryo kind of like should be in the end. At that time, there's not a lot of embryo. That's why you need this marking. And I'll just go back here. And then he used the flow fields to just kind of like here organize and reorient these guys. So he didn't use the green signal, he only used the flow fields I showed you. And you can see quite nicely in the end of the day, you can register these guys, and we have here kind of like this biological confirmation. So with this at hand, we can now do kind of like hopefully the overlay of many measurements with like all of these beads, so that we can actually get a visual representation of the forces involved there and then match that with the flows. But this is something that we're doing now that's going on. Okay. It's a general problem, as I said, like we would like to relate flows and forces in tissues. And this is something that is also a very important problem in cancer. And this is really something I will also rush through a little bit, but it's a pretty nice project by Shweta, where we ask the question like, can we maybe by looking at the flows here, learn something about the forces driving cells into a surrounding tissue? And of course, like, I mean, this is a very important question, and we know already that tumors have different pressure inside them. They also exert pressure on the surrounding. We also have kind of like quite a change in morphology that's based on ECM rigidity. We've seen beautiful talks about this. And we also know that in many systems we have switches of invasion modes by cell-cell and cell-ECM interactions. So we were particularly interested in kind of like flows that are generated here. And for example, here, if there's something that's driving it by pressure. So what we do is we have spheroids that we generate like by an agarose bed, and we put these in a capillary, and then we can kind of like mount the capillary in the light sheet microscope. And what you will then see is something like this later. We have here only the nuclei marked. So these are just like tracer beads to know what's going on the outside. These are nuclei marks. And just to show that this is not kind of like in thin air, all of this would be the imaging here of the collagen around it. So in reality, our spheroids are more like little pancakes. This is what they look from the side, but you can now very nicely look at this. And you can also take a movie over time. You can then look at things like mean square displacement of the motion here. This is typically subdiffusive. And this is something that happens. These were HeLa cells, by the way, so there's no real invasion happening here. This is not a cell line that's known to be invasive. Now, we had here quite high collagen concentration of 2.5 milligram. And then the question was, can we actually make here outgrowth happening? And what Shweta actually did, she played with different collagen concentration. I would like to show you the very thin collagen concentration that we did. This is only 0.5. This is a very thin collagen, very soft. And what happens there is kind of mind-blowing, because it's the same situation. Everything is the same. The only difference, the concentration of the collagen. And now what happens, this is 12 hours, we see these amazing bursts outwards. And what's happening here, this was really for us dazzling. We wanted to understand that. And it's really just the low collagen concentration. So this is kind of like putting them next to each other. This is the 2.5 and this is the 0.5 milligram. So what's going on here? And again, the idea was to figure out what's the physics of it and what are the forces here. And so what we looked at was kind of like, is there kind of collective migration happening here? And just to like already pitch for the next talk here, you can ask the question, like, to which extent do these guys kind of like work together? These are ants like moving here through sand. And the question that we wanted to ask you, to which sense do we have here something like cooperativity or like collective motion? And a way to quantify this is normally something like a correlation length that you can calculate. Basically, that tells you how far away cells are still moving in a similar fashion. And there is a certain length scale that you can calculate here by just looking at these correlations. And that what you can see here over time, this is the moment when we have the bursts. Indeed, this correlation length, like first it goes down. That means just the cells are getting more random. Suddenly we have a burst in this correlation length. So that suggests that the cells are indeed doing something collectively. And then we thought like, oh, cool, okay, collective means they talk to each other by forces. So we inhibited then ECAT, and we expected that this is going away, and it wasn't. So really, the collective migration we see here doesn't have to do anything with like cells touching each other or knowing each other. And that was a moment when we thought, like, okay, there's not so much left over. It can only be something like pressure. And to kind of like see that, what we did is we were trying to measure the volume of these cells. Unfortunately, we only had the nuclei, so we actually did the cheap version. We did a Voronoi expansion here. We looked at then, this is the center of the cell. These are kind of like these interactions with the Voronoi model. And out of this, you can get an estimate of the volume. And what you see in this 0.5, in this very thin, in this exploding spheroid, you see that the volume actually increases drastically. And that tells us already that we probably have here something like a volume effect. I will kind of like skip this almost. So we wanted to confirm this by having, this is not a chewing gum, the idea was here to have an image of the spheroid, and we have here an elastic surface, so we actually put cells in there, and we wanted to measure how much, this is side view now, how much this is actually pushed down, and that gives you an idea about the pressure generated, and you can indeed see this again here. This is the confocal cut, right? When it gets dark, it just means it's pushing down. So we see that these systems are pushing down, and now if I just show that very quickly here, this is in 0.5 milligram. This is the gel that explodes in our situation here, the collagen. And these are measurements where we have no collagen or we have the higher collagen, and you see hardly any indentation here. So that basically suggests that really have just an increase in pressure that is provoked by the volume increase. We can talk later about where that comes from, but this is a kind of like a nice, it's maybe a less exciting final outcome, but it shows quite nicely how you can really use physics, imaging, and a systematic approach here to understand something about the forces driving these systems. Just for the people who might care, this is this confiner that I just showed you. It's kind of neat because you can go down to something like a couple of microns here. And here, for example, these are neutrophils. This is traction force of them that are just squeezed in here down to a couple of microns. You can do traction force because we have these polyacrylamide gels here. This is actually the C component of the traction force here. Okay, this is just the eye catcher, right? This looks beautiful. This is a very, very thin, like we're only talking about 4 microns again here. This is the nucleus here, and actin is in red. All right. So for the last, how much time do I have? 5 minutes? Okay, perfect. So for the last minutes, I would like to show you something that is also, this came a lot out of something that in French is called bricolage, right? So we were just fiddling around and we were trying to solve things. And now actually that even turned into a little company. And I just, my disclaimer here, I'm part of that startup. But the main idea of this is actually not to become rich, but to provide these kind of chambers to the field, because I couldn't ask my workshop to produce them all the time for all the people in the world. But it's a nice thing, and I will show you in a second how it works. So the idea, yeah, well, I will first show the problem, or like what has been done so far. So the idea is rather simple. If you want to learn something about muscle tissue, right? You can grow nowadays muscle tissue in vitro, like organoids, quite nicely. That works really very well. But the problem typically is that if you do this just in a bulk situation, that the muscles don't really well align. And to overcome that, what you typically do is you actually have some kind of like attachment points. For example, here this was really done by Velcro. You grow these cells first in the dish, and then you put them in a protein scaffold, and then they self-organize ideally and form something like this. This is actually a muscle here. Let me run the video. This will twitch in a second. Yes, boom, here it is. So this is kind of cool because you can, with this, really very nicely actually start asking forces about muscle organization. It's not a real muscle, right? Because it misses all the developmental features, but it's a very nice model system. It's much better than just looking at them in a 2D situation here. So there are many kind of like ways of doing this. The one common problem is actually you need to, for example, have them here. You have here this kind of kink in there, and the idea is that otherwise when you have such a post and the cell kind of like is grown here, it will just slip off. So people have done really some very nice engineering to get that solved. And what you can do with these kind of like the systems, you can, for example, look here at a DMD. That's a real we have very, very severe muscular dystrophy, and you can measure, for example, the fourth generation. You can see that healthy cells, and typically the DMD cells are much weaker. There's one outlier here, but typically the DMD cells are weaker. So you can recapitulate what you find in vivo in these systems. Now, one problem of these systems is typically that it's very hard to do good imaging with them, because you can see here they're typically far away from the substrate, and that was really where this chamber idea came into place, and that's kind of like the show-off thing, like having people that know Blender is quite cool. So what you see here is this top, and in the end of the day, we have these posts, and here there's basically the tissue, and then when the tissue contracts, you see the post moving, and knowing the stiffness of the posts, right, you can calculate the forces. Now, this is how that would look like. The posts are very stiff because all of this is made out of plexiglass. It's a big advantage over PDMS because it doesn't absorb molecules. And then you see there's, it's not a lot, but there's a tiny motion here, and then Matthias actually came up with a cloud-based nice program where you just drag and drop your images, and then you get kind of like here these forces. So you can also kind of like have electrodes and twitch them, you know, and then what Matthias here, for example, quite nicely shows that he's training them here over time. And this is just like each of these colors is later in time. We start here right after starting. All of this is done after 2 weeks in culture, by the way, right? So you put these cells in culture, you let them organize for 2 weeks. This muscle needs to fuse, it needs to form. And then his time point 0 is just when he starts pacing. And pacing means he gives them a shock every 2 seconds. So he just makes the muscle work. And you can see quite nicely already after something like 20 hours here, we have a strong increase in the forces. And that's exactly now what triggers my attention, because I would like to know what happens here. Is this something that happens inside the cells? Is it a reorganization? Is it an expression of proteins that we see here? So these are all the kind of questions that I love to look at now. So this is just to show you how that kind of like self-organizes. That's kind of nice. You just put these cells in there. And then what is shown here is basically that you can also have some of these calcium dyes in there. And we have very nice microscopy because we're so close to the substrate. This would be a tetanus contraction, which is really just giving it for like kind of like 10 seconds, a very strong signal. And the cool thing about that is actually also that you can have these beautiful images now. So this is really the tissue in the chamber, like just the chamber put on a spinning disk microscope, something like 15 microns imaged, and then we have here a projection. What you see here is actually a fusion of 2 cells, right? So for the experts, you can ask me now a lot of important questions like what proteins are there? Is it really a fusion? We haven't looked at this. This is really just like playing with it. What you see over here, that's also kind of neat. You see really there's already a myotube because all of these nuclei, they're kind of like going, sorry, I'm too quick. They're just going together here. So this is a lot of fun now. And we're really starting to look deeper into this. But I mean, the take-home message of that I would like to give you, if you care about like having good microscopy of such muscle systems, because other systems, very often you have to unmount them later and you have to put them back into the microscopy situation. And that works. This is not a problem, but it's extremely tedious. And very often you actually start to damage the tissue. So the advantage here is just like you have it there, you put it on the microscope and you go for it. All right. So then we also tried to do a little bit of science with this. And this is something that was really surprising. So looking at what's done in the field, we thought, okay, let's use our chamber to get forces of these DMD patients, of the cells from patients that suffer from the muscular dystrophy. And what you see here is, again, just like such a contraction every now and then, yeah? And here you see how these posts move. And this is just an image of these cells. You can hopefully see the striation here. And this is just from the DMD patient. You can already see the tissue is heavily affected. And if you now measure just these contractile forces, in blue are the healthy ones. These are 2 different kind of like regions of the body. This is like from the back and this is from the legs. But you can see that overall we very nicely recapitulate that the diseased cells are weaker. So far, so good. So we were happy. But then we thought about another measure that we will get, because while these things kind of like generate, they also generate like a little pulling force during their contraction and kind of like the generation of the tissue. And we call this the homeostatic tension. So this is the tension that the system has, even if it's not triggered, right? Because it kind of like formed that structure. There's mechanical force in it. So we just measured that, and we were of course expecting that the DMD cells are also weaker there. But it was very surprising to see that actually the DMD cells are systematically stronger. And that is very interesting because it suggests that there is a signaling problem in these cells for the kind of like for the mechanoregulation to kind of like put in the right force or like arrange the right force for the muscles. And we have now the hypothesis that what happens in DMD is that this dystrophin gene or dystrophin protein, which is a monster, is also a mechanosensor, and this is something that we work on. So that's pretty cool. You can also anti-correlate that. That means like the higher actually your kind of like homeostatic tension that you build up while you grow, the lower is your contractile potential. So this is really something that we look at now. So pretty cool system. You can have a lot of fun with it, but you can also drive it further. And that's really now the work of here. Till is, by the way, a stellar PhD student who's now a postdoc, so he stays a bit in the lab, who has started most of this, and now Paula is taking over. So if you remember, if I go back, and if you see this, you might see that, well, here, these posts are extremely bright. We're illuminating it from the top, right? So why are they so bright? Well, it's actually quite simple. These posts are nothing than light guidance. They have a higher refractive index, right? So they just funnel the light down. So we thought, like, why not using this? And again, like Khalil said this, we would like to get rid of the microscope here. It's the wrong meeting. But the idea is just to be more efficient and quicker. So the idea is just putting some infrared LEDs up here and have some position sensitive detectors down there. And then we can actually put this is how that looks like. Again, like bricolage, right? So 3D printed, like done in the workshop, all homemade here, a lot of fun. You see here these detectors. And then in here, you see this is the chamber actually just sliding in there. So we just slide the chamber in there, and then you have here are the LEDs, and you have here these detectors. You know, the cool thing is what you can do, and this is really Paula's job, is actually to start generate iPSC generated cardiomyocytes, and they just beautifully, because they contract on their own, so you don't actually have to trigger them. And this is a little bit sped up. Normally these guys contract with something like every second or every other second. And now this is the first data, and that really is kind of like 3 weeks old. What I show you here, this is really this guy sitting in the microscope and just looking at it, and you see one post going up here, the other post goes down, right, because they do this. And then now you can kind of like just quantify nicely the peaks and these kind of like first points here, and then this is an overlay over all of these kind of like 11 cells. And now we start playing a lot with different drugs to see like if we can actually learn something about these curves, and would like to see to which extent it really behaves like a living muscle. And in the last minute, or am I dead then? 30 seconds, just to show off. So this is now Anna, and she's actually now adding here piezo elements. So this is a piezo element. These are the light-guided guys. And with this piezo element, you can move one of the posts now. So this is here just doing rheology with it. This post is moved. And you can do the same thing then later on the microscope or in the chamber. And this is the first data we get here. These are oscillations. And this would be the shear modulus. And this would be the loss modulus, like the elastic and the viscous part, directly out of a tissue. And this is just the comparison here between the PSD detection in the microscope, sorry, not in the microscope, in the incubator or in the microscope. And yeah, I do not do any overview. I just say thanks to an amazing group. We have a lot of fun, and I'm really blessed to have such good people. And I would like to thank you for your attention. And sorry.
Hello, thank you for this amazing presentation. It was really nice. Regarding the spheroid and the bursts,
yeah,
that was mind-blowing for me. What would determine the point of the burst? Like when you see which would be the determination, and do you think it would be related to the heterogeneous porosity, for example? Oh, yes. Yeah.
You're absolutely 100% right. I just kicked this out because I knew I'm over time. So it's actually this. So we were very excited because we thought we can learn something about the cells, their weak points and all of this. But then when we carefully looked at it, it's actually there are some void regions because the gel is so thin. And what happens is they just expand in these. So it's in the end it's less exciting, to be honest, right? Because
did you modelize it? Like, is there a model you think of doing about?
Yeah. So what we think is actually we have collagen in there. We have the receptors triggered, right? So the problem is just it's so soft that I think the cells try to pull on the environment, but there's no resistance, and then they become apoptotic. I think they're just like in the wrong environment, they become apoptotic, they swell, and the swelling is actually what we see here. So it's from a physics perspective still cool, but from a biomedical perspective maybe it's less great. But it's
thank you very much.
Yeah.
Any other questions?
It's too much. I'm sorry. Like I pushed in a lot.
Thanks for the nice talk. So maybe that's already what you did, and I didn't get it. But on the developing zebrafish embryo, it looks like the shape of the yolk sac can have some information. Like when you have the wave of cells propagating, it seems like it bulges out. Yeah, could you apply this Navier-Cauchy approach to the yolk sac and get some information?
Yes, you can, but we haven't done it yet. We're pretty much occupied by the inside. There is already some work on this, where people are actually looking at the yolk sac. It's, I think, much easier in that sense than the whole cells because it's not so dynamic, but there's quite some internal dynamic, actually, as you say, in the yolk sac. So there's some flows there too. And that was something that is not so easy to model. So we haven't done it yet, but I think you're absolutely right. One should look at this.
Thanks.
Thank you, Timo, for the nice talk. Your posts are not circular or in section.
Yes.
Is there a reason for that?
Yeah, actually, we machine them out of PMMA, out of plexiglass, right? And the problem is when you want to make them long, what happens is that they start vibrating and they break, so it's hard to machine. So what we did is we actually generated these ovals to make it stable enough to machine it, and in the direction where we need the kind of like lower stiffness. So we have the same cross-section, but we have this change, so we have a lower stiffness in that direction. That's why. That's kind of important. Yeah.
Thank you.
Yeah, thank you very much for your amazing talk.
Thanks a lot.
I was wondering with your tumor assay, your spheroid, it's usually monoclonal cells. Have you tried to do the same with tumoroids where you have higher heterogeneity? And in that case, do you have, I don't know, a collective migration of few cells based on their state that are going to only be able to migrate or to move?
Yeah, so awesome question. We actually started to team up with the clinic. But then we realized that in our new light sheet that we got in Goettingen, we have a hard time in making the cells or these clusters like survival enough. So we actually now switching to a situation, I also kicked that out, but we're using the chamber now. So we have in the chamber, we can use here fibroblasts to generate this matrix. And then in there, we can also add the cancer. So this is a cancer tumor now. But these are, again, like MDA and B. So this is not heterogeneous as you asked for, which is a great question. And then we can now start kind of like looking at this. The cool thing about this is it's at least it's directional. So you can see that there is a mechanical force and these cells move out. But we haven't done that. And I think it's an extremely interesting question. One could also try the other way around, having monoclonal cells and then get the ones that are out and trying to do a proteome or transcriptome of these.
Yeah, exactly.
Yeah. But no, we haven't done it.